Now, so we're going to go over some of the kind of key concepts here from what we talked about. I'm a fan of tables. I think tables are a good way to bring everything together and compare and contrast, and they are a great study tool for you to refer to, just as you're reviewing this later on. So I'm going to create a few tables here that should kind of summarize some of what we talked about and hopefully make it a little bit easier for you to review before the exam. Okay, so we're going to talk about absorption first. And we'll talk about the 3 macromolecules. We'll talk about the enzymes that break them down. We'll talk about the micronutrients that they're broken down into. And then finally, the mechanism by which they're absorbed. Okay, so we've got carbs first. And we know that carbohydrates are broken down by amylase. Let's put a note here that this starts in the mouth. So this is the only macromolecule where digestion starts in the mouth. The micronutrients that these must be broken down to in order to be absorbed are glucose or galactose. Okay, so if you had a really long branched polysaccharide, disaccharide, you can break it down to maybe limit dextrins, which we described, but it must be further broken down by the time it gets to the small intestine into glucose or galactose to be absorbed. The mechanisms for absorption we said across the apical surface. It was primary active transport, so sodium potassium pump, not sodium potassium, excuse me, secondary active transport. As well as on the basolateral surface, this is where we had primary active transport, as well as facilitated diffusion. Okay. So that was carbs and how carbs are absorbed. Let's look at proteins. So protein digestion starts in the stomach. And the enzymes involved are going to be pepsin in the stomach. Oops, let's get another color. So pepsin in the stomach, but then we also had quite a few other enzymes. So there was trypsin, chymotrypsin, and carboxypeptidase. Okay, and these were in the intestines. And remember, these had to be activated in the intestinal lumen via the enterokinase. Okay, so these were the activated forms. The inactivated forms that were released were trypsinogen, chymotrypsinogen, and procarboxypeptidase. Okay. All right, let's look at the micronutrients here. So these can be either peptides or amino acids. But let's make a note that they must be amino acids to cross the basolateral surface. So they can get into the cell as peptides, but in order to cross the basolateral surface, they must be further broken down within the cell. Okay, and then the mechanisms here were the exact same as carbs. They got into the cell via secondary active transport exchange for sodium, and then they can move across the basolateral surface via facilitated diffusion. Okay, and then the sodium-potassium pump is creating the relative gradients to facilitate that. Let's look at fat. So fats had a bit of a unique case because of their makeup, because they're nonpolar. A little bit different. So fats, we said, were broken down by lipase, but we needed bile salts to emulsify to smaller fat droplets. Okay, so fats could be broken down by lipase after those huge goblets were broken down into smaller droplets. They must be broken down into monoglycerides or fatty acids. And then once they get inside the cell, remember, they can cross the cellular surface via diffusion because they're fats as monoglycerides and fatty acids. But once inside the cell, there's a few things that happen. They were repackaged to triglycerides in the smooth endoplasmic reticulum, or the SR. And then they were also repackaged into chylomicrons in the Golgi apparatus. I'm going to put GA for Golgi apparatus. Okay, so there was a bit of repackaging, reprocessing that needed to take place of these simple fats once they get inside the cell. And then in order for them to be absorbed, they could move by exocytosis and diffusion. They could go right through that membrane into the lacteal. And remember, the lacteal is a lymphatic capillary. Okay, this is capillary. It's a bit jumbled. Okay, so that's how we absorb fats into the bloodstream, and that's how we absorb all of the nutrients here, okay? So this is a good way for you to kind of synthesize everything we talked about with the breakdown of these macromolecules, the enzymes involved, what they're broken down to in order to be absorbed, and then exactly how they move across the apical surface of that cell and the basolateral surface into the bloodstream. Alrighty, okay. Let's remind ourselves of the stomach cells. We kind of did this earlier, but let's do it here again. Again, because I'm a fan of tables. So let's look at the stomach cells and their secretions and their function. So we've got 4 stomach cells that we described: the chief cells, the parietal cells, the G cells, and the neck cells. Now, let's remind ourselves that these cells are located in the gastric pits, also called the gastric glands. Okay. All right, so let's look at the secretions from these cells, and then the function of those secretions. So the chief cells secrete pepsinogen. Okay, and the function of pepsinogen, we kind of just went over this, is to activate, well, first it's activated by hydrochloric acid of the stomach to pepsin. And then the function of pepsin is to break down proteins. Okay, so that's the function of pepsinogen. It's the inactivated form of pepsin, which breaks down proteins when it's activated by hydrochloric acid. Parietal cells secrete two things. They secrete hydrochloric acid as well as intrinsic factor. Okay, and these have two different functions. Acid had two functions as well. One is to activate pepsin for protein breakdown. And the other was as a part of the immune system, right? It's going to be a defense mechanism against pathogens, bacteria, viruses, etc. And that was the function of acid. Intrinsic factor was required to absorb vitamin B12. Okay. Let's look at the G cells. So the G cells are kind of straightforward. They only secrete gastrin, one of the main motility and secretion hormones of the GI. And gastrin has two functions. It's going to increase hydrochloric acid secretion of the stomach. It is also going to increase the stomach motility. Okay. And then lastly, the neck cells. The neck cells secrete mucus. And the function of mucus was to coat the stomach lining and protect against acidity. Okay, so to protect the underlying stomach lining from the acidity from the hydrochloric acid. Okay, so these are the function of the gastric pit cells, the cells that secrete them, and then what the exact secretions of each cell is. All right, let's look at one last table, and then we're going to move on to some checkpoint questions and a few practice multiple choice questions. So the final table here is just all of the hormones that we just spoke about that regulate motility and secretion. So so the GI regulatory hormones, we talked about gastrin. We talked about CCK. Okay, so we'll talk about where that hormone is secreted, what the stimulus for its secretion is, and then what the action is. So gastrin, let's start off with gastrin. Gastrin is secreted in the stomach. We just talked about where the G cells of the gastric pits, so the stomach. In brackets, I'll put G cells. The stimulus for gastrin release is the presence of proteins or protein products, so proteins, peptides, or amino acids, as well as the physical distension of the stomach is another stimulus here. Okay, the action of gastrin, right, that's going to be hand in hand with stomach secretion and stomach motility. So we're going to have increased hydrochloric acid secretion, and we're going to have motility. We're going to have motility of two areas. We're going to have increased stomach motility and ileum motility. Okay, so it's not only going to get the first part of the GI, the stomach kind of moving, but it's also going to get the distal part of the GI moving to kind of move things along. This is why sometimes right after you eat, you have to use the bathroom, right? It's going to get the distal part of the GI moving as well, the motility moving there. It's also going to relax the ileocecal sphincter, which is moving intestinal contents down into the colon. Okay, so that's gastrin. That's one of the first hormones. We also talked about CCK. So CCK, or cholecystokinin, the location for this is the duodenum and jejunum. The stimuli here are going to be the presence of fats or protein. When we say protein, this could also include peptides or amino acids, which are those breakdown products. The function of CCK is to block acid secretion, so it's going to block the function of gastrin, right? It's going to have an opposite effect here. Block acid secretion. As well as block stomach motility. Okay. It's also going to stimulate the gallbladder and the pancreas. To release their secretions of bile and enzymatic fluid. Gallbladder, pancreas, enzyme, bile secretion. It's going to increase that. And then lastly, it's going to relax the sphincter of Oddi. Right, that sphincter that's guarding the opening into the duodenum, it's going to relax that so that bile can enter, so that these pancreatic secretions can enter as well. Let's look at secretin. And remember, secretin is potentiating the effects here, so they're kind of pretty much the exact same effects of secretin and CCK, but its stimulus is going to be different. The stimulus here is going to be hydrochloric acid in the duodenum. Should have been in the duodenum. And then the exact same location, so duodenum and jejunum. Oops. And this is secretin. Okay, so secretin and CCK have a synergistic relationship. They're bringing about the same response, but they're doing it by different stimuli. And then the last one that I'll squeeze down here, we didn't talk about this too much, but we did mention it, so I'll go ahead and put it here. This is GIP, glucose-independent insulinotropic peptide, and that is secreted by the duodenum and jejunum as well. The stimulus is distension of the duodenum. So duodenal distension. And the action is to inhibit acid secretion and motility. And then the other important function is to secrete insulin from the pancreas. Oops. Hope we can see that there. Okay. So it's going to be stimulated by the distension of the duodenum, and it's going to inhibit or block acid secretion, but increase the insulin secretion from the pancreas. Okay. So these are our 4 important hormones. We mainly focused on these 3. We didn't speak about this so much, but just so you have an idea of where it falls in with the others. Okay. So let's now look at some checkpoint questions here that I think we skipped over. And then I have a few practice multiple choice questions. But let me see if there are any questions first before we do that. So as far as the GI, make sure you have an understanding of each of the parts of the GI, what their function is. There's a really nice summary table in that first packet where you can easily see those functions and add things that may be missing. Make sure you have an understanding of the absorption, that first table we drew, how those macromolecules are broken down, what they must be broken down to, how they are absorbed. Have an understanding of the major hormones that influence the GI system, and then those 2 reflex pathways and the 3 areas of control, the cephalic, the gastric, and the intestinal phases of control, and the long and short reflex pathways. Okay, so those are some of the major